Hello, welcome again. Today we're going to look at chapter 9 on the organization of the human genome. It starts off talking about some comparative complexity of the genome between the human genome and the mitochondrial genome. This is the human genome, but this is not the mitochondrial genome. If you look closely, and you'll find that one there, hang on, that is the mitochondrial genome, right? So look closely. So it just says that comparatively, that is the proportion of the human genome, and that is the proportion of the mitochondrial genome. Although in the cells, there's normally just one copy of the genome. Well, a diploid will have two copies, but there are many mitochondria. Then the book talks about well, I wanted to read you through the book, but then it becomes monotonous and boring. So I think you're gonna have to do the reading on yourself, mind you. But you need to be able to ask me questions if you don't understand any part. So what I'm going to do, I'm just going to go through very quickly in terms of what are the take-home message from some sections of this book. So this it starts off talking about some of the current understandings of the human genome. It talks about the surprises that most of the human genome is being transcribed. I mentioned this class, and this has given birth to the knowledge on RNA genes, and then also some non-coding RNA transcripts. Section 9.1 talks about the size. Well, it focuses on, it says on general organism human genome, but it zoomed in on the mitochondrial genome. 16.6 kilobases. It's a single type of circular double-stranded DNA. It talks about there are the two strands of the circular mitochondrial DNA identified as the heavy strand and then light strand. The heavy strand because it has a lot of guanines. I asked you in class, why are they heavy? and and then it talks a bit about how it replicates, focusing on the presence of what is called as D-loop. D-loop means displacement. We've seen this bit in when we talk about model of recombination, right? If you can't remember, you have to just go through again. You'll see that there is a part where you talk about strand invasion and displacement. And this is also the area where the DNA is sometimes referred to as a triple helix. The next subsection talks about the mitochondrial genes and the transcription. There are 37 genes altogether, and 28 of them are on the heavy strand, and then 9 of them on the lighter strand. There's a concept of multigenic transcript. If you remember operons, you know how they are transcribed into one piece. 3 genes, if you remember, like Z, 3 introns like Z, Y, and A are transcribed as one transcript, and then they are and then expressed. Okay, so in this case, did I say in this case? I say in this case, the transcripts are transcribed, and later the mature RNA are going to be cleaved. A little bit about the structure of the genes. All 37 mitochondrial DNA genes lack introns. And then it goes into the subsection on mitochondrial genetic code. Now, this table shows the for most function requires contribution of the nuclear genome. It just says that while the mitochondrial genome has 37 genes, it has its own tRNA and it has its own ribosomal genes, but the other gene, the structural genes, come from the nuclear genome. So here, for the different purposes, for the different functions of the mitochondria, you can see the contributions of the nuclear genome. The mitochondria has 60 codons. I asked you in class, what is the number of codons for the human genome, right? If you can't remember, you have to start counting. Instead of 3 in the normal human genome, the mitochondrium has 4 stop codons, and these are it. The UAA, UAG, and then AGA and AGG are stop codons in the mitochondria, while UGA, which is a normal stop codon for a human genome, is actually it actually encodes for tryptophan. What else? Okay, here it says there are 60 codons, right? It mentions up there there are 60 codons, but there are only 22 different RNA genes. This part of the lecture, or this part, this phenomenon, you've seen this before in perhaps in your second year, where we talk about the wobble hypothesis. Lagerquist, what was it? Crick, I think. They did. They were trying to answer this question and came up with the third wobble hypothesis. You see, as I mentioned also in class, the codon anticodon interaction is based on hydrogen bondage between hydrogen bonds between um, the codon and anticodon, but because the GC has 3 hydrogen bonds each, so they are expected to be that only 2 bases are required compared to the AT, in theory. Did I just do that on video? Okay, this is the comparative analysis, or just looking at the difference between the human nuclear and mitochondrial genomes. These are again just facts, just to show you the sizes, the number of genes, and nothing spectacular to be seen here. And then it goes into talking about the human genome, nuclear genome content. It's 3.1 gigabase. That's a haploid genome. There are 24 different chromosomes. All right, not for all gender. Think about it. I mentioned this in class. Then it talks about euchromatin and heterochromatin. And of course, if you remember heterochromatin, you've got the constitutive as well as facultative. It explains later somewhere in here. It says certain chromosomes, notably 1, 9, 16. Okay, here. Certain chromosomes, notably 1, 9, 16, and 19, also have different amounts of heterochromatin in the euchromatic region, have significant amounts close to the centromere, and this is also called the pericentromeric region. The last 2 paragraphs talks about the GC content. It has about 41% average, but it ranges. That's the trend that you're going to see for a lot of the different aspects of the human genome. They try to describe what to expect, but then again, talking about the number of genes, from the very small to the very big, the different sizes of chromosomes, the GC content, they're all very variable to us. There are some similarities here and there, but in general, there's considerable variation. Now, for this part, the number of human genes, I'm gonna deal with that in the next video. All right, so I'll see you in the next video. Bye.